Look at your hand. Spread your fingers wide and hold them up in front of you. 5 fingers. 4 spaces between them. Open air, where your hand ends and the world begins. That separation feels like the most natural thing in the world. It was not always there. 7 weeks after you were conceived. Your hand looked nothing like this. It was a paddle, a flat, unbroken sheet of tissue with no separation between one finger and the next. The hand that would eventually learn to hold a pen, play an instrument, reach for another person in the dark, was a single continuous surface. Everything connected, nothing distinct. What changed between that paddle and the hand you are looking at right now was not growth. It was not the addition of anything. What happened was that somewhere between week 7 and week 8, roughly 150,000 cells received a message. The message was simple. It was, in the most direct biological sense possible, an instruction to die. And they did, quietly, in order. Those cells did not fight the instruction. They did not malfunction or get attacked or suffer any kind of damage. They executed a program written into the DNA of every cell in your body. A program so old, it predates the existence of hands by hundreds of millions of years. And they disappeared. The space between your fingers is the record of 150,000 deaths that happened exactly on schedule so that you could hold a cup, turn a doorknob, wave goodbye. Your hand was sculpted by absence. Those cells did not die because something went wrong. They died because something went extraordinarily right. There is a system inside every cell of your body whose entire purpose is to end the cell that contains it under the right circumstances. Not accidentally, not as a last resort, as a designed function, as deliberate as a heartbeat, as necessary as breathing. Evolution did not just select for cells that could live. It selected for cells that knew when to die. That is what this story is about. Not death as tragedy, not death as the enemy of life. Death as the price of becoming something more than a single cell floating alone in an ancient ocean. Death as the condition that made complexity possible. Death as the darkest and most necessary upgrade in the 4 billion year history of life on this planet. The question is not why we die. The question is why life built death into itself on purpose. Go back 2 billion years, and the Earth is alive, not alive the way it is now, with forests and oceans full of fish and a sky crossed by birds, alive in a quieter, stranger, more fundamental way. Every surface that touches water is covered in thin sheets of microbial life. The ocean teems with single cells, dividing and competing and dying by accident and dividing again. From space, the planet would have looked almost the same as it does today in outline. A blue world, a wet world, a world clearly doing something. But zoom in and the picture changes entirely. Every living thing on Earth at this moment is doing exactly one thing. It is trying to become 2 living things. Division is the entire project. A cell absorbs nutrients, copies its DNA, splits down the middle, and produces 2 cells where there was one. Those 2 cells do the same thing. The goal is simple enough to state in a single sentence: persist and replicate for as long as conditions allow. There is no cooperation in the way we understand cooperation. There is no specialization. There is no cell that decides to be a heart cell while another decides to be a brain cell. Every cell is its own complete organism, carrying all the machinery it needs to survive, making all its own decisions, answering to nothing outside its own membrane. In this world, The death of a cell is the worst possible outcome. Full stop. If you are a single-celled organism and you die, the game is over. Your entire existence ends at the moment your membrane fails and your chemistry dissolves into the surrounding water. There is no body to carry on. There is no offspring that benefits from your sacrifice. There is only you. And then there is nothing. So, for a very long time, every pressure of natural selection pointed in one direction. Live longer. Divide faster. Resist damage. Repair errors. Keep the membrane intact. Survive. This is the world that life built over its first 2 billion years. A world of extraordinary biological diversity at the chemical level, with cells running dozens of different metabolic strategies, breathing different gases, living at different temperatures, filling every available niche in the ocean and the sea floor and the thin, wet surfaces of emerging land. But for all that chemical diversity, the fundamental logic was the same everywhere. Every cell was playing the same game. Every cell was playing alone. Then something happened that would eventually change everything about that game. It did not happen all at once. But somewhere in that ancient microbial world, the first tentative experiments in cellular cooperation began. Cells that stayed near each other after dividing. Cells that developed the ability to pass signals from one to another. Cells that began to behave as if the group mattered. Most of these experiments failed. The pressure to defect, to stop cooperating and start competing for resources, was enormous. A cell that cheated the group and prioritized its own replication would out-reproduce the cooperators around it. Cooperation is always vulnerable to this. The math of natural selection makes defection attractive in the short term, even when it is catastrophic in the long term. But some experiments held. In some environments, groups of cells found ways to maintain cooperation across divisions. They began to develop crude forms of specialization. They began to behave, in the most primitive sense, like organisms rather than collections of individuals. And right there, at the very beginning of that transition, a problem appeared that nobody could have anticipated. A problem so fundamental that it would shape the entire subsequent history of complex life on Earth. A single cell playing alone has one interest: its own survival. Every chemical pathway, every molecular machine inside it is oriented toward that single goal. But a group of cells trying to function as a unit has a different interest: the survival of the unit. And those two interests are not always the same. A cell that divides when the unit needs it to stop is not being strategic. It is being itself. It is following the only instructions it has ever had. Instructions written into its biology by 2 billion years of evolution that said, survive and replicate above all else. The problem of multicellularity is not a problem of bad actors. It is a problem of old instructions meeting new situations. And here is the deepest version of that problem. If cells are going to cooperate in a body, if they are going to specialize and coordinate and serve functions beyond their own replication, then the body needs a way to control them. It needs a way to tell a cell when to divide and when to stop. It needs a way to tell a cell when its existence has become a liability rather than an asset. It needs a way to tell a cell to die. The question the next several 100 million years of evolution would work to answer was not how to keep cells alive longer. It was how to make death useful. Evolution did not just select for cells that could live. It selected for cells that knew when to die. Imagine trying to build a city where every citizen has been raised for their entire life, with one absolute rule: look out for yourself, compete for everything, trust no central authority. This is not a political statement. This is a description of 2 billion years of cellular evolution. Every cell that has ever lived learned to survive under exactly those conditions. The winner was always the cell that replicated fastest, consumed most efficiently, and resisted death longest. Now, take 37 trillion of those cells and ask them to build a human being. The scale of what multicellular life had to accomplish is genuinely hard to hold in your mind. A human body contains somewhere around 37 trillion cells. Those cells perform thousands of distinct functions. Neurons carry electrical signals. Red blood cells transport oxygen. Muscle cells contract. Pancreatic cells produce insulin. Immune cells identify and destroy pathogens. Liver cells process toxins. Each of these cell types has given up the generalist survival strategy of its single-celled ancestors and committed to doing one specific thing for the benefit of a body it cannot see and does not understand. That commitment has a cost that rarely gets discussed. A neuron that specializes completely into a brain cell cannot divide easily. It has traded replicative flexibility for functional sophistication. A red blood cell goes even further. It ejects its own nucleus, discarding its DNA entirely, to make more room for the hemoglobin that carries oxygen. It becomes, at the moment of full maturity, a cell that has made itself incapable of reproduction. It has accepted, at the molecular level, a kind of permanent sterility in service of a function that keeps the body alive. This is extraordinary when you stop to think about it. These cells are not making sacrifices in any conscious sense. They have no awareness of what they have given up. But the biological reality is that specialization requires restriction, and restriction is the opposite of everything 2 billion years of single-celled evolution selected for. So, how does a body maintain this? How does it keep 37 trillion cells, each one carrying the full genetic instructions for unrestricted division? Each one descended from ancestors that survived by doing exactly that, in line. How does it prevent any one of them from deciding, at the molecular level, to stop cooperating and start competing? Part of the answer is communication. Cells in a body are embedded in a constant conversation, receiving signals from neighboring cells, from the bloodstream, from the nervous system, that tell them where they are, what they should be doing, and whether they should be dividing. A cell that receives the right signals behaves as expected. A cell that loses contact with those signals begins to behave unpredictably. But communication alone is not enough. Communication can be ignored. Signals can be misread. And in a body that is replicating its DNA trillions of times every day, errors accumulate. Mutations happen. A cell can develop the ability to misinterpret signals, or to stop receiving them altogether, and continue dividing anyway. Communication keeps the cooperative body running under normal conditions. It is not robust enough to handle every failure. What the body needs, in addition to communication, is enforcement. And enforcement, in a biological system, has a very specific meaning. It means that a cell which has stopped cooperating, which has lost the ability to respond to the signals that keep it in line, which has become a liability rather than an asset to the whole, needs to be removed permanently, in a way that does not cause additional disruption to the surrounding tissue. This is the impossible problem of multicellularity. How to remove cooperation failures before they cascade into something that destroys the whole system. How to build an enforcement mechanism into every single cell in the body. An enforcement mechanism that can be triggered from outside the cell or from inside it, that acts reliably, that leaves no mess, that scales across 37 trillion individual units without central coordination. The solution that evolution found is apoptosis. And the reason it took so long to evolve, the reason the gap between the first multicellular experiments and the first complex animals spans hundreds of millions of years, is that apoptosis is not a simple mechanism. It is an interlocking system of molecular checks and counterchecks, triggers and brakes, signals and suppressors that has to work reliably in every cell type in the body, under every condition, without false positives that kill healthy cells, and without failures that allow damaged cells to survive. Building that system from scratch, through the blind accumulation of random mutations, each one either improving reliability or being discarded, is one of the most difficult problems in the history of life. But life had something that no human engineer has ever had. Life had time, hundreds of millions of years of time, and an almost incomprehensible number of organisms to test variations in, and the merciless filter of survival to keep only the versions that worked. What emerged from that process is the system running inside your cells right now. A system so well calibrated, so deeply integrated into the basic operations of every cell in your body, that most people live their entire lives without once thinking about it. It runs beneath consciousness. It runs beneath sensation. It runs in the dark interior of cells that have no idea what they are part of, executing decisions that keep the whole system running. It is, in the truest sense, the invisible infrastructure of your existence. There are two ways a cell can die. The first way is the way most people imagine death at any scale. Something goes wrong from the outside. The cell is starved of oxygen, or attacked by a pathogen, or exposed to a toxin, or physically crushed by injury. The membrane ruptures. The contents of the cell spill out into the surrounding tissue. Neighboring cells are suddenly bathed in molecules they were never meant to encounter. Signals that say, something has gone catastrophically wrong here. The immune system responds. Inflammation spreads. The tissue swells and heats and becomes painful, as the body mobilizes everything it has to deal with the mess. This kind of death is called necrosis. It is death as emergency, death as system failure, death that announces itself loudly and forces everything around it to stop and respond. The second way a cell can die looks nothing like that. A cell receives a signal. The signal might come from outside, from a neighboring cell or a circulating molecule in the bloodstream, or it might arise from inside the cell itself, from sensors that monitor the integrity of its DNA or the state of its mitochondria. The signal does not cause immediate collapse. Instead, it initiates a sequence of events so orderly, so precisely coordinated, that describing it as death feels almost inaccurate. It feels more like a carefully executed plan. The cell begins to shrink. The cytoplasm condenses. The chromatin inside the nucleus begins to compact into dense, defined structures along the edges of the nuclear membrane. The DNA itself is cut, not randomly, not by damage, but at specific sites between genes, by enzymes that know exactly where to make each cut. The nucleus fragments. The cell continues to pull inward, rounding up, becoming smaller and more defined, even as it dismantles itself from the inside. Then the membrane changes. On the inner surface of a healthy cell membrane, there is a molecule called phosphatidylserine. In a living cell, it stays on the inside. It is actively pumped inward, kept away from the outer surface by proteins that maintain this asymmetry continuously, spending energy to do so every moment of the cell's life. But in a cell undergoing this process, those pumps shut down. phosphatidylserine flips to the outer surface. It appears on the outside of the cell like a flag, and every immune cell in the vicinity knows exactly what that flag means. The dying cell is packaging itself. It buds off small, membrane-wrapped parcels, each one containing fragments of DNA, fragments of organelles, fragments of the protein machinery that kept the cell running. These parcels are called apoptotic bodies. They are tidy, sealed, clearly labeled packages that the immune system can recognize and engulf without triggering any inflammatory response. The contents never spill into the surrounding tissue. The neighboring cells never know anything went wrong. The immune cells arrive, consume the packages, and the space where a cell used to be is simply empty. No inflammation, no damage signal, no disruption to the tissue around it. The cell is gone as if it was never there. This process is called apoptosis. The word comes from ancient Greek, chosen deliberately by the scientists who named it in 1972. It means the falling of leaves from a tree in autumn, or the dropping of petals from a flower. Not violent loss. Natural, seasonal, inevitable release. The kind of ending that is part of the system rather than a failure of it. Now, think about the molecular machinery required to make this happen. At the center of apoptosis is a family of proteins called caspases. They are proteases, meaning they cut other proteins at specific sites. Caspases are not active by default. They exist inside every cell in an inactive form, waiting. They are a set of loaded mechanisms that are permanently on safety, stored in every cell, available instantly if needed, but incapable of firing on their own. What releases the safety is a cascade. An initiator caspase gets activated first, either by signals from outside the cell or by signals from the mitochondria inside it. That activated initiator caspase then activates executioner caspases. The executioner caspases begin cutting. They cut structural proteins that hold the cell's shape. They cut proteins that normally keep the DNA bundled safely. They activate the enzymes that fragment the DNA at those precise sites. They dismantle the cell from the inside with a specificity that no random chemical process could replicate. The mitochondria play a central role in this. Mitochondria are the energy-producing organelles of the cell, the structures that convert nutrients into the chemical fuel that powers everything the cell does. They are also one of the primary sensors that monitor whether the cell should continue to exist. When a mitochondrion detects that the cell is too damaged to repair, or that it has received signals indicating it is no longer needed, it releases a protein called cytochrome C into the interior of the cell. Cytochrome C is normally confined inside the mitochondrion, doing its job in the energy production chain. When it appears outside the mitochondrion, floating free in the cytoplasm, it is a signal that cannot be misread. It means the mitochondria have made their assessment, and when that assessment points toward death, the caspase cascade begins. Standing between the mitochondria and the final decision is a family of proteins called BCL2, named for the B-cell lymphoma gene, where they were first discovered. The BCL2 family contains both pro-death and pro-survival members, and the balance between them determines whether the mitochondria release cytochrome C or hold it back. Some members sit on the outer mitochondrial membrane and block the release. Others punch holes in that membrane to force it. The balance between these two factions, constantly shifting in response to signals from inside and outside the cell, is the mechanism by which the cell integrates all available information about its own condition and the needs of the body around it, and translates that information into a binary decision, live or die. The elegance of this system is not just biological. It is architectural. The decision is made at multiple levels simultaneously. Signals from outside the cell, signals from the cell's own DNA damage sensors, signals from the mitochondria. Each of these feeds into the same cascade. Each of them can trigger the process independently if the situation is severe enough. And each of them can be overridden by survival signals if conditions change. The system is not a simple switch. It is a continuously updated calculation running in real time, weighing the cell's condition against the body's needs. Always capable of revision until the moment the executioner caspases begin their work. After that point, there is no reversal. The commitment to die, once made at the molecular level, is total. The cell does not get to change its mind when the caspases begin cutting. And this irreversibility is not a design flaw. It is a design feature. A system that could be reversed after initiation would be vulnerable to the very kinds of molecular errors and corruptions that the system exists to eliminate. Once the decision is made, it has to be final. What apoptosis represents, in the deepest sense, is the solution to the problem that Chapter 2 described. The body needs a way to remove cells that have become liabilities. And it needs to do this without damaging the surrounding tissue, without triggering immune responses that cost more than they solve, and without relying on any single point of failure. Apoptosis solves all three requirements simultaneously. It removes the cell cleanly. It packages the remains for effortless disposal. And it does this through a redundant, multi-pathway system that cannot easily be defeated by a single mutation. Every cell in your body is carrying this system right now. The caspases are loaded and waiting. The BCL2 family members are balanced on their respective sides of the mitochondrial membrane. Cytochrome C is doing its normal job inside the mitochondria, ready to become a death signal if anything pushes the balance too far. This is not a morbid thought. It is the reason you are alive. In the 1960s, a British scientist named Sidney Brenner was looking for a problem worth solving. He had already made his mark. He had contributed to the work that cracked the genetic code, helping establish that three-letter sequences of DNA spelled out the instructions for building proteins. That was not a small achievement. But Brenner was restless in the way that the best scientists tend to be restless, always looking for the next question that nobody had thought to ask yet, or that everyone had thought to ask, but nobody had figured out how to answer. The question he settled on was deceptively simple. How does a fertilized egg become an organism? Not in the biochemical sense, not the chemistry of gene expression, but in the physical sense. How does a single cell divide and divide and divide until the cells know where to go and what to become? How does a cell in what will become the head know it is in the head, and a cell in what will become the tail know it is in the tail? This question was completely unapproachable in anything as complicated as a mouse or a fly. Too many cells. Too much going on at once. You could never follow the full story from beginning to end. So Brenner chose a worm. Caenorhabditis elegans is not impressive to look at. It is transparent, about 1 millimeter long, lives in soil, eats bacteria, and under normal circumstances attracts no attention from anything. Brenner chose it for one reason that turned out to be perhaps the most important methodological decision in 20th century biology. The worm is transparent, meaning you can watch every cell inside it under a microscope without cutting it open. And the worm, it turned out, has a fixed number of cells, not approximately fixed, not fixed within a range, exactly fixed. Every adult hermaphrodite C. elegans has exactly 959 somatic cells. Every single worm. Every single time. Around the world. In every laboratory that has ever grown these animals. The number is the same. 959. This was already remarkable. But what John Sulston discovered when he began mapping the complete cell lineage of the worm, tracking every cell division from fertilized egg to adult under the microscope, hour after hour, day after day, was something that stopped him cold. The worm did not arrive at 959 cells by producing exactly 959 cells. It produced 1,090 cells, and then 131 of them died. Not approximately 131. Exactly 131. Every worm. Every time. Always the same cells. Always in the same order. Always at the same point in development. Sulston watched them die. He sat at his microscope and observed individual cells. Cells whose entire lineage he had tracked from the moment of the first division of the fertilized egg, reach a specific point in development and undergo a specific change. They condensed. They became refractile under the microscope, meaning they changed the way they interacted with light. And then they were gone. The neighboring cells closed in around the space they had occupied. And if you did not know where to look, you would not know anything had been there. The deaths were not accidents. They were not caused by anything going wrong in the environment or in the worm's development. They were part of the developmental program, as precisely specified as the divisions that produced the cells in the first place. The worm had a schedule of deaths built into its developmental instructions, and it followed that schedule with a reliability that no biological process of this kind had ever been shown to possess. This was new, not slightly new, fundamentally new. The prevailing assumption in biology at the time was that cell death in development was a passive process. Cells that did not receive enough survival signals simply fading out, not a process actively driven by the cell itself. What Sulston was looking at said something different. It said that some cells were programmed to die, that the program was written into the genome of the organism, and that it executed with a precision that could only mean it was being selected for, that it mattered, that organisms whose developmental deaths were less precise left fewer descendants than organisms whose deaths were exactly right. Robert Horvitz took the next step. If these deaths were programmed, there had to be genes running the program. Horvitz set out to find them. What he found was a small set of genes, with names like SED3 and SED4 and SED9, that controlled whether cells lived or died. Worms with mutations in SED3 did not execute their programmed deaths. The cells that should have died survived, and those worms were not healthier for it. They were subtly wrong, their development slightly off, their bodies carrying cells that had no business being there. Worms with mutations in SED9 went the other way. Cells that should have survived began dying. The worm fell apart. The system required both. Genes that drove death and genes that suppressed it, balanced against each other. The outcome determined by which side won in any given cell at any given moment in development. When Brenner, Sulston, and Horvitz received the Nobel Prize in Physiology or Medicine in 2002, the committee described their work as revealing a new world. That is not hyperbole. What a 1 millimeter transparent worm living in soil had revealed was that programmed cell death was not an anomaly. It was a fundamental feature of how complex life develops and maintains itself. And the genes they found in the worm were not unique to the worm. The same basic machinery, modified and elaborated, runs in every complex animal on Earth. SED3 is the worm version of the caspases described in the previous chapter. SED9 is the worm version of BCL2. The same system, the same basic logic. Running in a worm with 959 cells and in a human body with 37 trillion. Because the logic was worked out so early in the evolution of complex life, and worked out so well, that evolution never found a reason to replace it. 131 cells. Always the same cells. Always the same schedule. That precision is a message. It says that the body does not leave death to chance. It plans for it. It builds it in. It specifies which cells will die, and when, and in what order, writing those specifications into the genome just as carefully as it writes the specifications for which cells will survive and what they will become. Your development was not just a story of cells being born and growing and finding their place. It was also a story of cells being born in order to die, fulfilling their developmental function in the moment of their disappearance, shaping the organism they were never going to be part of. The worm showed us that, and once you know that, you cannot look at a developing body the same way again. Go back to your hand. You looked at it at the beginning of this story, the space between your fingers. The 150,000 cells that died on schedule so that your fingers could separate and become distinct. That felt like a striking fact when you first encountered it. Something to hold on to while the bigger story built around it. Now you have the bigger story. You know what apoptosis is. You know the caspases and the BCL2 family and the cytochrome C release and the apoptotic bodies packaged so neatly for disposal. You know about the worm and its 131 deaths that happen the same way every time. You know that this system is not an accident or a byproduct. It is infrastructure. Now, look at your hand again with all of that in mind. What happened in week 7 and week 8 of your development was not a simple erasure. It was a coordinated wave of apoptotic signaling that swept through the interdigital tissue with spatial precision that no human surgeon could replicate. The cells that died were not randomly distributed. They occupied specific positions, specific zones between the developing finger rays, and they received death signals at specific times in a specific sequence that determined not just whether the fingers would be separate, but how separate they would be. How long each finger would end up relative to the others. How the web of skin at the base of your fingers would be shaped. Errors in this process are not hypothetical. When the apoptotic wave fails to complete in the interdigital zones, fingers fuse. Syndactyly, the condition in which 2 or more fingers remain connected by tissue, results from exactly this, from programmed cell death that started but did not finish, or that was never fully initiated in certain zones. The hand that should have been sculpted remains partially uncarved. The hand is the most visible example. It is not close to the most dramatic one. Consider the brain. The human brain begins developing early in gestation and does not finish until well into the 3rd decade of life. Over that entire period, it is producing neurons at a rate that would seem, if you only looked at the production side, almost reckless. The developing brain generates far more neurons than it will ever use. In the period of peak neuronal production, the brain is creating cells faster than it could possibly wire them all into functional circuits. Then it starts cutting. Between birth and early childhood, the human brain loses somewhere between 50 and 70% of the neurons it produced during development. 50 to 70%. More than half the neurons your brain ever made are gone before you are old enough to form lasting memories. They died through apoptosis, triggered by a mechanism that is as elegant as anything in biology. A neuron survives if it successfully connects to its target. If it receives enough survival signals from the cells it is trying to reach to keep its BCL2 balance tipped toward life. A neuron that fails to make sufficient connections does not receive those signals. Its internal balance tips the other way. It initiates its own death. This is called synaptic competition. Neurons compete for survival signals from their targets. And the neurons that win are the ones that make the best connections. The ones that lose die. The result is a brain wired by competition, in which every surviving circuit has proven itself against alternatives that were tried and discarded. The brain you have today is not the brain you were born with. It is the brain that survived the elimination of the brain you were born with. Learning, in the most literal biological sense, is partly a story of cell death. This principle, that complex structures are built by eliminating excess rather than by adding exactly what is needed, runs through the entire body. The kidney tubules that filter your blood were formed partly by apoptosis, creating hollow spaces within solid tissue. The tubes of the inner ear, the semicircular canals that tell your brain which way is up, required programmed cell death to hollow them out during development. The lens of the eye, which has to be transparent to function, is made of cells that have undergone a modified form of programmed death, eliminating their nuclei and organelles to become clear, losing everything that made them conventional cells in order to serve a function that conventional cells cannot perform. The chambers of your heart were separated and refined by apoptosis. The valves that keep blood moving in one direction were shaped by it. The spaces in your joints, the cavities in your sinuses, the architecture of your digestive tract, all of these carry the signature of death, working as a construction tool. There is a concept in sculpture that speaks to this. Some sculptors describe their work not as adding material to create a form, but as revealing the form already inside the stone, removing what does not belong until only the essential shape remains. The body works something like this. Except, the sculptor is not an artist with a vision. The sculptor is a molecular cascade, triggered by signals that encode spatial and temporal information across millions of cells simultaneously, coordinated not by any central intelligence, but by the same kind of distributed chemical communication that runs every other biological process. There is no blueprint being followed consciously. There is only a system of signals and responses that, through hundreds of millions of years of refinement, reliably produces a body shaped correctly enough to function. The absences are as important as the presences, the spaces as important as the structures, the deaths as important as the divisions. Your nervous system illustrates this at a level that becomes almost philosophical when you follow it far enough. Every memory you have formed in your life involved not just the strengthening of certain synaptic connections, but the weakening and elimination of others. The process of learning is inseparable from the process of forgetting, and forgetting, at the cellular level, involves the pruning and sometimes the elimination of connections that are not being reinforced. Your brain is continuously editing itself, continuously removing what is not needed to make room for what is, continuously using a form of controlled cellular death to refine the wiring that defines who you are. The person you are right now is partly the result of neurons that died in exactly the right place at exactly the right time. You are not only the sum of everything you have experienced and remembered, you are also the sum of what your brain decided to let go. And this is where the story of apoptosis begins to shift from development into maintenance. Because the body is not a finished product. It is not a sculpture that gets completed and then sits unchanged. It is a dynamic system that requires continuous renewal, continuous removal of cells that have aged past their usefulness or accumulated damage that makes them dangerous, continuous replacement of those cells with fresh ones. The average cell in your body does not live as long as you do. Red blood cells live about 120 days before they are removed from circulation and replaced. The cells lining your intestine turn over every 3 to 5 days. Skin cells live a few weeks. Liver cells live somewhere between 1 and 2 years. Even the cells that seem permanent, the neurons you use every day, are maintained through processes that require constant molecular upkeep, constant removal of damaged components, constant decision-making about which parts of which cells should be kept and which should be cleared. Apoptosis is part of this ongoing maintenance in ways that extend well beyond development. A cell that has accumulated too much DNA damage becomes a candidate for apoptosis. A cell that has been infected by a virus becomes a target for immune-mediated apoptosis. A cell that has simply reached the end of its functional lifespan, that can no longer perform its job reliably, begins to receive signals that tip the plateau balance toward death. The body is continuously making these decisions, trillions of them every day, mostly invisibly, mostly successfully. You have never felt a single one of them. That is how well the system works. Your immune system is an army that has never seen the enemy. It was built to fight. Every pathogen your immune cells are prepared to recognize. Every bacterial protein and viral surface molecule and fungal antigen that your white blood cells can identify and respond to is a threat that your body has either encountered before or that your immune system has been primed to recognize through vaccination. The army is built from experience. It learns. But here is the problem, that every army built from learning faces. Learning requires trying things that do not work. And in an immune system, trying things that do not work means producing cells that are prepared to attack the wrong target. The immune system generates its diversity through a process of controlled randomization. The genes that encode the receptors on T cells and B cells, the molecules these cells use to identify their targets, are assembled during development by a process that randomly combines gene segments to produce receptors with an enormous range of possible binding profiles. The system does this because no genome is large enough to pre-specify receptors for every pathogen the immune system might ever encounter. Rather than trying to predict every possible threat, the body generates a huge library of random receptor configurations and then selects from that library the ones that are useful. This produces millions of different T cells and B cells, each one carrying a receptor with a different binding profile, each one prepared to recognize a different molecular target. And some of those receptors, inevitably, will recognize the body's own proteins. This is not a flaw in the system. It is a mathematical certainty. If you generate enough random receptor configurations to cover the space of possible pathogens, some of those configurations will match molecules that are already present in the body. The randomness that gives the immune system its breadth necessarily includes self-reactive cells as part of the output. A self-reactive immune cell is not merely useless. It is one of the most dangerous things a body can produce. A T cell that recognizes the proteins of the body's own nervous tissue, and that is allowed to activate and proliferate, will attack the nervous tissue the same way it would attack a pathogen. The body cannot distinguish from the inside between damage done by an external threat and damage done by its own immune system. The inflammation is the same. The destruction is the same. The outcome, if the attack is severe enough and sustained enough, is the same. The body solves this problem the only way it can. It kills the dangerous cells before they can do damage. The place where this killing happens is the thymus, a small gland sitting just behind your sternum that is largest and most active during childhood and gradually shrinks as you age. Immature T cells migrate to the thymus from the bone marrow and undergo a process that has no gentle name in immunology. It is called thymic selection, and it is, in functional terms, an elimination tournament from which most participants do not emerge alive. T cells that enter the thymus first have to prove they can recognize the molecular markers that identify cells as belonging to this body. Cells that cannot do this are useless. They cannot tell self from non-self, cannot participate in any immune response, and they die. The cells that pass this first test then face a second one. They are exposed to a library of the body's own proteins, presented by specialized cells in the thymus called medullary epithelial cells, which have the unusual ability to express proteins from many different tissues. Proteins that would normally only be found in the pancreas, or the eye, or the nervous system. A T cell that reacts too strongly to any of these self-proteins, that shows signs of the kind of activation that would be appropriate against a pathogen, but is catastrophically wrong against the body's own tissue, is identified, and then it dies through apoptosis, right there in the thymus, before it has ever left to enter circulation, before it has ever had the chance to cause the damage it was capable of causing. The numbers are striking. Somewhere between 95 and 98% of the T cells that enter the thymus never leave it alive. Most of them fail one of the two selection tests. They either cannot recognize self, or they recognize self too well. Only the 2 to 5% that thread the needle, recognizing the body's own markers without reacting to the body's own proteins, are allowed to mature and enter the bloodstream. 2 to 5%. The thymus is not generating a population of immune cells with a modest attrition rate. It is conducting a near total elimination, keeping only the rare cells that meet an extraordinarily precise specification. The immune system that circulates in your body and protects you from infection is the survivor of a culling that would look, if you described it in any other context, like catastrophic failure. It is not failure. It is the only way to make an immune system that is dangerous enough to kill pathogens without being dangerous enough to kill the body it lives in. The violence has to be educated, and the education requires death. When this process works correctly, the self-reactive cells are removed. The surviving cells circulate and monitor for genuine threats, and the body maintains the balance between immune vigilance and immune restraint that keeps you healthy. When it works correctly, you never think about it. The thymus does its quiet, lethal work, and the immune system that emerges from it is calibrated to fight everything that is not you. When it does not work correctly, the consequences are familiar, even if the underlying mechanism is not. Rheumatoid arthritis is the immune system attacking the lining of the joints. Multiple sclerosis is the immune system attacking the myelin sheath around neurons. Type 1 diabetes involves immune attack on the insulin-producing cells of the pancreas. Lupus involves a systemic immune attack on multiple tissues simultaneously. These are not diseases of immune deficiency. They are diseases of immune misdirection, of self-reactive cells that survived the thymic selection they should not have survived, that entered circulation carrying receptors tuned to the wrong target. Each of these conditions begins at the molecular level with a failure of apoptosis. A cell that should have died in the thymus did not die. The mechanism that would have eliminated it did not execute correctly. One cell survived that should not have, and then it divided, and its daughters divided, and the army that was supposed to protect the body began turning on it. There is something in this that cuts deeper than biology. The immune system has to be capable of tremendous violence to do its job. It has to be able to recognize a threat and destroy it completely, rapidly, without hesitation. That capacity for violence is not an unfortunate side effect of how the immune system works. It is the central feature. An immune system that hesitates or holds back is an immune system that lets infections establish themselves, lets viruses replicate unchecked, lets bacteria overwhelm tissues before any response can stop them. But a system capable of that kind of violence, turned against the body it lives in, is catastrophic. The thymus is the institution that teaches the immune system to be violent in the right direction. And apoptosis is the mechanism the thymus uses to enforce that lesson. The cells that learn it survive. The cells that do not learn it die in the classroom before they can cause harm in the world. Most of the time, this works. Most of the time, the education holds. The immune system circulating in your body right now has passed its test. It knows what it is supposed to attack and what it is supposed to leave alone. Most of the time, the cases where it does not work are not rare enough to be theoretical. Millions of people live with autoimmune conditions, with immune systems that passed an imperfect version of the thymic test, that carry self-reactive cells that survived when they should not have, that are slowly waging wars against their own tissue without any understanding that the enemy they are fighting is themselves. Every one of those cases begins with the same event, invisible and unremarkable at the time it happened. A cell in a thymus, somewhere in childhood or before birth, failed to initiate apoptosis when the signal came. The signal arrived. The BCL2 balance shifted. The caspase cascade was supposed to begin. It did not, and the cell lived and divided. And eventually the consequence of that one missed death became a disease, with a name and a diagnosis and a lifetime of management. This is what it means to say that apoptosis is not optional. It is not a backup system or a fail-safe that the body runs when things go wrong. It is primary infrastructure. It is as essential to the immune system's function as the receptors that identify threats or the signaling pathways that trigger attacks. Without it, the immune system cannot be trusted with the power it has. The dark education is not a metaphor. It is a process. It runs in the thymus of every mammal that has ever lived. It selects through death. It enforces through death. It produces, when it works, an immune system capable of enormous violence directed with extraordinary precision. When it works, every cell in your body carries the same DNA. The neuron in your prefrontal cortex and the cell lining your stomach and the muscle fiber in your left calf all contain the same genome, the same complete set of instructions that was present in the fertilized egg you started as. What makes them different from each other is not what genes they have, but which genes they express, which instructions they follow and which they leave dormant. A liver cell is a liver cell because it activates the genes relevant to liver function and suppresses everything else. It has the instructions for being a neuron. It simply does not read them. This is one of the most remarkable features of multicellular life, and also one of its most profound vulnerabilities, because those suppressed instructions are still there. The capacity for unrestricted division that every cell inherited from its single-celled ancestors, the ancient biological imperative to replicate without limit, is present in every cell in your body right now. It is not gone. It is not deleted. It is suppressed, held in check by layers of molecular regulation built up over hundreds of millions of years of evolution. Regulation that is stable under most circumstances and fragile under some. Cancer begins when that suppression fails, never all at once. Cancer is not a sudden event. It is a process of accumulation, of small failures, building on each other over months or years or decades. Each one moving a cell slightly further from the cooperative behavior that the multicellular contract requires, and slightly closer to the ancient single-celled logic of replicate at any cost. The first mutation might affect a gene that controls how often the cell divides. The cell starts dividing a little more frequently than it should. The second mutation might affect a gene involved in DNA repair, so that subsequent mutations accumulate faster. The third might disable one of the checkpoints that normally pause cell division to check whether the DNA has been copied correctly. The fourth might affect a survival signal pathway, making the cell slightly more resistant to the signals that would normally push it toward apoptosis when things go wrong. Each of these changes, on its own, might not be enough to produce a fully malignant cell. The body has redundant safeguards. Lose one checkpoint and others remain. Acquire one pro-survival mutation, and the apoptotic machinery can still be triggered through other pathways. Cancer requires the accumulation of multiple independent failures, typically somewhere between 5 and 10 significant mutations, before a cell has dismantled enough of the cooperative infrastructure to become genuinely dangerous. This is why cancer is predominantly a disease of aging. Given enough time, given enough cell divisions, given the continuous low-level DNA damage that comes from metabolism and radiation, and the simple mechanical stress of copying 3 billion base pairs over and over across a lifetime, the probability of any given cell accumulating the right combination of mutations climbs. Cancer is not bad luck in the way that a car accident is bad luck. It is the statistical consequence of maintaining a complex biological system over decades. But among all the safeguards that have to fail for cancer to develop, the failure of apoptosis is the most consistently critical. Every cell in your body is continuously monitored for signs that something has gone wrong. DNA damage sensors scan the genome between divisions. Signals from neighboring cells check whether the local tissue environment is appropriate. Immune cells patrol for molecular markers of cellular dysfunction. The mitochondria monitor the cell's overall metabolic health. Any of these surveillance systems, when it detects a problem severe enough, feeds into the Blackall-2 balance and tips it toward death. The caspases activate. The cell is removed before the problem it represents can propagate. A cell that has lost the ability to undergo apoptosis has cut the wire to its own self-destruct mechanism. It can accumulate damage without the damage triggering the response that would normally eliminate it. It can receive death signals from neighboring cells or from the immune system and ignore them. It can sit in a tissue environment that is telling it to stop dividing and keep dividing anyway, because the mechanism that would enforce that signal has been dismantled. The protein most often involved in this dismantling has a name that, once you know what it does, carries a weight. Its simple label does not suggest. It is called P53. P53 is a transcription factor, a protein that binds to DNA and controls which genes get read. When DNA damage is detected in a cell, P53 is activated. It can pause cell division to allow time for repair. It can activate DNA repair pathways directly. And if the damage is too severe to repair, it can activate the genes that tip the cell toward apoptosis. P53 is, in the most functional sense, the guardian of the genome. It is the protein that stands between a cell with damaged DNA and the proliferation of that damage into daughter cells. P53 is mutated or inactivated in more than half of all human cancers. More than half. In some cancer types, the rate is higher. Colorectal cancer, lung cancer, ovarian cancer, head and neck cancers all show P53 mutations at rates that make it the single most commonly altered gene in human cancer. When p53 is gone, the cell has lost its primary mechanism for deciding that DNA damage is severe enough to warrant death rather than continued division. The guardian is gone. The cell divides with damaged DNA. The daughters inherit that damage. The damage accumulates across generations of cells in a way that, with p53 present, would never have been allowed to happen. The failure of apoptosis in cancer reaches beyond p53. The BLCL2 family itself is a target. In certain lymphomas, a chromosomal rearrangement places the BCL2 gene under the control of a highly active promoter, causing the cell to produce far more of the pro-survival BCL2 protein than normal. The balance in the mitochondrial membrane tips dramatically towards survival. The cells become resistant to the death signals that would normally eliminate them. They persist. They accumulate. They form the masses of slowly proliferating cells that define certain leukemias and lymphomas. Cancer has found multiple independent ways to defeat the same system. This is not a coincidence. It reflects the fact that apoptosis is not one thing that cancer has to overcome. It is a system with multiple entry points and multiple execution mechanisms. A natural selection operating on cancer cells finds and preserves mutations that defeat it by any available route. Cancer cells that happen to acquire an apoptosis-defeating mutation survive longer and divide more than cancer cells without that mutation. They become the dominant population. They pass the mutation to their daughters. The tumor evolves toward apoptosis resistance the way any population evolves toward any advantageous trait. This is what makes cancer so difficult to treat. A chemotherapy drug that kills cancer cells by triggering apoptosis will also, over time, select for the cancer cells most resistant to apoptosis. The treatment is a selection pressure. The cancer population evolves in response to it. The tumor that comes back after treatment is often more resistant than the tumor that was there before. Not because any individual cell changed in response to the drug, but because the cells most capable of resisting the drug were the ones that survived while the others died. Understanding this has shifted how oncologists think about treatment. Rather than trying to kill every cancer cell, which gives the surviving resistant cells a competitive advantage in a cleared landscape, some strategies now aim to restore the function of the apoptotic pathway the tumor has learned to circumvent. Drugs called BH3 mimetics work by binding to the pro-survival BCL2 proteins and blocking them, restoring the balance in the mitochondrial membrane toward death in cells that have tipped it too far toward survival. One of these drugs, venetoclax, has shown remarkable effectiveness in certain leukemias, where BCL2 overexpression is the primary driver of cell survival. It is, in the most direct sense, a drug that works by giving cancer cells back the ability to die. But step back from the clinical detail and look at the larger picture. Cancer is what happens when a cell remembers how to live like its ancestors lived before there were bodies, before cooperation, before the contract. It is not a foreign invasion. It is a cell that has, through the accumulation of mutations, reverted to an earlier mode of existence, one where the only rule is divide and survive, where the signals from the body around it are noise rather than instruction, where the mechanism that was supposed to enforce cooperation has been dismantled piece by piece until nothing is left to enforce it. The cell that becomes cancer is not doing anything new. It is doing something very old. And the body's primary defense against it is the same system whose origin and function this entire story has been tracing. When apoptosis works, cancer cells are eliminated before they can accumulate the additional mutations they need to become dangerous. The body produces cells with damaged DNA constantly, every day, in every tissue. Most of those cells never become cancer because the apoptotic program runs correctly. The guardian protein activates, the balance shifts, the caspases begin their work. The cell disappears, cleanly, without inflammation. And the tissue continues as before. The cancers that develop are the ones where this failed, where the guardian was mutated before it could respond, or where the balance was already tipped so far towards survival that the death signal could not overcome it, or where the execution mechanism was blocked before it could run to completion. Every cancer that has ever existed began with a cell that should have died and did not. A cell stopped dying when it should have, and kept dividing when it should not have. The contract had been broken from the inside. You are not the same person you were 7 years ago. Not in the philosophical sense, though perhaps in that sense too. In the literal, physical, cellular sense. Most of the cells that make up your body right now did not exist 7 years ago. They are replacements, produced through division from cells that were themselves replacements from cells before them, in a continuous chain of renewal that has been running since before you were born. The body is not a fixed structure. It is a process, a continuous act of self-replacement in which old cells are retired and new cells take their place, maintaining the architecture and function of tissues across decades of use, in a world full of things that damage biological molecules. This renewal is one of the most fundamental features of living systems, so fundamental that it rarely gets noticed. Operating beneath awareness, the way breathing operates beneath awareness. Essential and invisible. Apoptosis is part of this renewal. Cells that have aged past their functional prime. Cells that have accumulated too much damage to repair reliably. Cells that have reached the end of the replicative lifespan written into their biology receive death signals and are eliminated. New cells divide to replace them. The tissue continues. But here is the question this chapter is about. If renewal is continuous, if apoptosis is continuously removing old and damaged cells, and fresh cells are continuously replacing them, why do we age at all? Why does the renewal process not simply keep up forever, maintaining the body at peak function indefinitely? The answer involves evolution. And it is deeply uncomfortable in the way that evolutionary answers to questions about human experience often are. Evolution does not care about you after you have reproduced. This is not a statement about value or meaning. It is a statement about the logic of natural selection. Natural selection acts on reproductive success. Traits that increase the probability of survival and reproduction before and during reproductive years are strongly selected for. Traits that cause problems only after reproduction, after the period when genetic contribution to the next generation has already been made, face much weaker selection pressure. Genes that are harmful in old age but neutral or beneficial earlier in life can persist in populations for exactly this reason. The selection pressure against them is weak because they cause their damage after the critical window has closed. This creates a situation that the biologist Peter Medawar described in the 1950s, and that has been elaborated and refined ever since. The genome of every complex organism contains a kind of accumulated debt, genes and gene variants that are useful early and harmful late, or that were never selected against because the harm they cause falls outside the window where selection is strong. Aging is partly the payment of this debt. The late expression of genetic costs that natural selection never had strong enough reason to eliminate. Apoptosis is caught in this logic from multiple directions simultaneously. The first direction is cell senescence. When a cell has divided too many times, when the protective caps at the ends of its chromosomes have eroded past a critical threshold. It stops dividing. This is a safeguard against cancer, because cells that cannot divide cannot accumulate additional mutations through division. But senescent cells are not neutral. They do not simply sit quietly in the tissue taking up space. They secrete a cocktail of inflammatory molecules, growth factors, and matrix-degrading enzymes that alter the tissue environment around them. In small numbers, this secretory behavior has beneficial effects, promoting wound healing and tissue remodeling. In large numbers, accumulated over decades, it contributes to the chronic low-level inflammation that underlies many diseases of aging. The body can clear senescent cells through apoptosis. Immune cells identify them and trigger their elimination. But this clearance becomes less efficient with age, for reasons that appear to involve both a decline in immune function and changes in the apoptotic sensitivity of senescent cells themselves. The senescent cells accumulate faster than they are removed. The inflammatory environment they create worsens, and that worsening environment feeds back into the apoptotic machinery of neighboring cells in ways that are not always beneficial. The second direction is even more direct. Apoptosis, when it runs in the wrong place or at the wrong rate, does not repair the body. It damages it. The nervous system is the most vulnerable to this, because neurons are among the cells least capable of replacing themselves. Most tissues can regenerate from populations of stem cells that divide to produce new functional cells. The adult brain has very limited capacity for this. The neurons you have in your cortex are mostly the neurons you will have for the rest of your life. They can remodel their connections. They can strengthen and weaken synapses. They can compensate for local damage through plasticity. But they cannot be replaced in significant numbers if they are lost. When apoptosis runs incorrectly in the nervous system, the losses are permanent. In Alzheimer's disease, neurons in regions critical for memory and cognition undergo apoptotic death at rates that the brain cannot compensate for. The triggers involve the accumulation of toxic protein aggregates, the amyloid plaques and tau tangles that are the pathological signatures of the disease. These aggregates interfere with mitochondrial function, shifting the Bl2 balance toward death in neurons that are not damaged beyond repair. That would continue functioning if the aggregates were not present. That die not because they have failed, but because the environment around them has been poisoned. The result is a progressive loss of neurons in regions that define the person. Memory goes first, in most cases. Then language. Then the ability to recognize faces. Then the capacity for complex reasoning. Then the basic coordination of bodily functions. Each step is neurons dying that should not be dying. Apoptotic machinery, running in the right cells at the wrong time, triggered by the wrong signals. Parkinson's disease follows a similar logic in the neurons that produce dopamine. Their high metabolic activity, making them uniquely vulnerable to the same imbalance. The pattern repeats across neurodegenerative diseases because the vulnerability is the same neurons that cannot be replaced, dying through apoptosis, triggered by the wrong signals at the wrong time. This is the slow price. Not a single payment, but a continuous accumulation of small miscalibrations, small failures of the apoptotic machinery to run correctly in every context, small excesses and deficits that individually are tolerable and collectively are not. The joints that ache, the memory that reaches for a word and comes back empty, the reflexes that are no longer quite as fast, the immune surveillance that no longer catches every damaged cell before it can accumulate another mutation, the slow dimming of systems that kept everything running. Aging is not apoptosis failing completely. Aging is apoptosis, slowly losing the calibration that made it work. And here is the part of this that matters most for understanding what the system is and what it is not. There is genuine scientific interest in interventions that might slow or reverse aging by targeting the apoptotic and senescent cell machinery. Drugs called senolytics aim to clear accumulated senescent cells, reducing the inflammatory environment they create and potentially restoring tissue function. Research into BCL2 family modulation explores whether shifting the apoptotic balance in specific tissues might protect neurons without increasing cancer risk elsewhere. Studies of organisms with unusual longevity, certain species of bat and naked mole rat, look for variations in apoptotic regulation that might explain their extended health span. This research is real, and some of it is promising, but it exists within a constraint that no intervention can fully escape. The apoptotic system was not designed for longevity. It was selected for reproductive success. The same features that make it effective at preventing cancer in early life make it difficult to preserve neurons in late life. The same calibration that produces a functional immune system in youth produces an inflammatory senescent cell burden in age. Evolution did not optimize for the experience of being 80 years old. It optimized for the experience of being 30, of being healthy enough to reproduce and to care for offspring until they could fend for themselves. And it accepted whatever happened after that as outside the selection calculus. We are living in the after. We are living past the window that evolution cared about, in bodies that were calibrated for a different era, with apoptotic machinery that begins to lose its precision at exactly the ages when modern medicine has made it possible for us to be alive. The mismatch between what the system was selected for and what we are asking it to do is not a failure of biology. It is a consequence of success. The success of medicine in extending lifespan has created a situation that evolution never anticipated, because evolution never needed to. The price of complexity was always going to be paid eventually. The apoptotic system deferred that payment across a lifetime. It kept the body running long enough to reproduce, long enough to raise children, long enough to contribute to the social structures that gave those children a better chance. And then it began, slowly, to lose the thread. The slow price is not a punishment. It is the other side of a deal that gave you everything you are. Step outside and look at a tree. Any tree. The one in your yard. Or the one at the edge of the parking lot. Or the row of them lining a street you walk down without thinking much about what they are doing. Pick one and look at it for a moment before reading further. That tree is not a passive object. It is a system of roughly 37 trillion cells, roughly the same order of magnitude as a human body, each one carrying out specific functions, each one embedded in a communication network that coordinates growth and defense and resource allocation across an organism that cannot move, cannot run from threats, cannot do anything except stand in one place and manage whatever the world brings to it. And inside that tree, right now, cells are dying on purpose. The leaves you see on a deciduous tree in summer are not permanent structures. They are seasonal investments. The tree produces them in spring at significant metabolic cost, uses them through the growing season to capture sunlight and fix carbon. And then, as days shorten and temperatures drop in autumn, initiates a process of controlled dismantling that is as precisely coordinated as anything in animal biology. The tree does not simply abandon its leaves. It dismantles them from the inside out, recovering as much as possible before releasing them. Chlorophyll is broken down, and the components are transported back into the branch for storage and reuse. Nitrogen, phosphorus, and other nutrients are pulled out of the leaf tissue and returned to the plant before the connection is severed. What remains when the leaf finally falls is a depleted husk, stripped of its most valuable components, returned to the soil to decompose. The separation itself is a programmed event. A layer of specialized cells, called the abscission zone, forms at the base of the leaf stem. These cells undergo changes in their cell walls, loosening the connections between them, until the physical attachment between leaf and branch weakens to the point where wind or gravity completes the separation. The abscission zone cells do not survive this process. They are sacrificed to facilitate the clean release of the leaf. This is programmed cell death in plants. The molecular executioners are different from those in animals. The caspases are not there. The BCL2 family is not there in the same form. But the functional logic is identical. A signal initiates a coordinated program of cellular self-dismantling that serves the survival of the larger organism at the cost of individual cells. Plants use programmed cell death throughout their lives in ways that reveal how universal the underlying logic is, independent of the specific molecular machinery that implements it. The xylem, the tissue that transports water from roots to leaves throughout the plant, is made of cells that have undergone programmed death as part of their maturation. A functional xylem vessel is a hollow tube, and it is hollow because the cell that formed it digested its own contents and then died, leaving behind only the reinforced cell wall that forms the conduit. The plumbing of every plant is built from the remains of cells that died to become pipes. The wood of a tree trunk is largely dead xylem, layer upon layer of cells that executed their developmental death program in sequence. Their empty, reinforced walls stacking up over years into the structural material that holds the tree upright. You have been sitting near, or beneath, or inside structures built from programmed cell death your entire life without knowing it. When a plant is attacked by a pathogen, a virus, or a bacterium, or a fungus, attempting to establish an infection, the plant responds with a strategy that reveals the same underlying logic that runs the animal thymic selection described in Chapter 6. The strategy is called the hypersensitive response. The cells immediately surrounding the site of infection receive signals triggered by the presence of the pathogen, and they die. Rapidly, deliberately, in a wave that spreads outward from the infection site and then stops. The logic is containment. The pathogen needs living cells to infect and replicate within. By killing the cells immediately surrounding the infection site, the plant creates a zone of dead tissue, a firebreak that the pathogen cannot easily cross. The sacrifice of a small number of cells at the perimeter protects the larger organism from an infection that could otherwise spread systemically and become lethal. The plant does not have a nervous system. It does not have an immune system in the animal sense. It cannot produce antibodies or activate T cells. What it has is the ability to instruct its own cells to die at the site of invasion. And that ability, stark and costly as it is often enough. Fungi tell a similar story. In filamentous fungi, certain hyphal cells undergo regulated death during development, hollowing out structures and shaping the architecture of the organism. The mushroom emerging from the ground is partly a product of cells that died in the right place at the right time, their deaths as necessary to its final form, as the deaths of interdigital cells are to a human hand. The molecular executioners are different. The logic is the same. None of this is apoptosis in the strict molecular sense. The specific proteins that execute programmed death in plants and fungi are not the same ones that run the process in animals. But the functional architecture is the same everywhere you look. Individual units dying in a controlled and programmed way to serve the survival and integrity of a larger system. the solution showing up again and again in lineages that have been separately evolving for hundreds of millions of years. This is the strongest possible evidence that the logic is not contingent. It is not something that happened to evolve in animals and then got borrowed by plants. It is something that any sufficiently complex biological system converges on, because it solves a problem that complexity inevitably creates. The problem is this: any system made of many individual units, each one capable of independent behavior, faces the continuous risk that the behavior of individual units will become misaligned with the needs of the whole. The more complex the system, the more ways this misalignment can occur. The more ways it can occur, the more important it becomes to have a mechanism for removing the misaligned units before their misalignment propagates or compounds. A single-celled organism has no use for programmed death because there is no larger system to protect. The individual unit and the whole are the same thing. But the moment you build a multicellular organism, or a colonial organism, or any system in which the behavior of individual units is supposed to be coordinated in service of collective function, you have created the need for enforcement. And enforcement, at the cellular level, means the ability to remove units that are not cooperating correctly. The plants found their version of enforcement. The fungi found their version. The animals found theirs. The molecular details differ. The evolutionary origins of the specific mechanisms differ, but the functional outcome is the same across all of them, because the problem they are solving is the same. Complexity requires the ability to subtract, not only to add, not only to grow and divide and accumulate, but to remove, precisely and on schedule, and without disruption to the surrounding system. Whatever has become a liability rather than an asset, what biology did, over hundreds of millions of years and across every major lineage of complex life, was find molecular implementations of this logic that were reliable enough, precise enough, and robust enough to maintain complex systems across the timescales that organisms actually live. The implementations are different because the evolutionary histories are different. The logic is the same because the problem is the same. And the problem, at its core, is the problem of being many things at once. A tree is many cells, trying to be one tree. A fungal colony is many hyphae trying to be one organism. A human body is 37 trillion cells, trying to be one person. In every case, the integrity of the whole depends on the controlled behavior of the parts, and in every case, the most powerful tool available for maintaining that control is the same: the ability of individual units to die for the whole. The machinery is different in every lineage. The signal is different. The execution is different. But the outcome, across every kingdom of complex life that has ever evolved on this planet, is the same. When it is needed, the cell dies, and the organism continues. Come back to the beginning. Not to the beginning of this story, though you will return there in a moment. To the beginning of life, to the ancient ocean, 2 billion years ago, where single cells divided and competed and died by accident and divided again, where every cell was playing alone, where the only rule was persist and replicate and nothing else mattered. Those cells did not know what they were. They had no awareness of the ocean they were floating in, or the planet they were part of, or the history they were making, by simply continuing to exist across billions of years of geological change. They were chemistry, organized, self-replicating, extraordinarily sophisticated chemistry. But chemistry without experience, without knowledge, without any sense of what they were building toward. What they were building toward, through no intention of their own, was you, not you specifically, not any specific complex organism, but the general direction of increasing complexity, of cells finding ways to cooperate that opened biological possibilities that solitary cells could never access. Nervous systems, immune systems, circulatory systems, the capacity to move through the world with coordinated purpose, the capacity to learn from experience, the capacity, eventually, to sit and read a story about your own origins and understand something true about what you are. All of that required something that the ancient single-celled world did not have and could not have had. It required a way for individual cells to die, not by accident, but by design. Not because they failed, but because the system they were part of needed them gone. The story of how that requirement was met is the story this entire account has been telling. It is the story of a molecular machine assembled piece by piece over hundreds of millions of years of evolution. A machine of extraordinary sophistication that sits inside every cell of every complex organism on Earth, loaded and waiting, ready to execute a program of self-dismantling so precise that it leaves no trace and causes no disruption. The machine is caspases and BCL2 and cytochrome C and P53 and 100 other proteins, whose names do not matter as much as what they collectively accomplish. What they collectively accomplish is control. Not control in the sense of restriction or limitation, though it involves both. Control in the sense of the difference between a crowd and a choir. A crowd is many individuals, each doing what they individually want to do. A choir is many individuals doing different things in precise coordination to produce something that none of them could produce alone. The difference between a crowd and a choir is not the number of people. It is the presence of a system that coordinates their behavior toward a shared outcome. That includes mechanisms for ensuring participation and for removing elements that disrupt the coordination. Apoptosis is part of what makes the body a choir rather than a crowd. It is the enforcement mechanism that keeps 37 trillion cells coordinated over decades of operation, in a world that continuously damages biological molecules and generates errors in replication and produces cells that have mutated away from cooperative behavior. Without it, the choir falls apart. With it, the body can maintain the coordination that makes thought and movement and sensation and all the rest of human experience possible. This is not a small thing. It is, in a very direct sense, the difference between the kind of life that was possible before programmed cell death evolved and the kind of life that became possible after, before single cells. Colonial clusters, the most primitive multicellular experiments. Nothing with a nervous system or a body plan or the capacity for the kind of sustained biological complexity that produces organs. The ceiling on what life could become was low, held down by the problem of cellular defection that multicellularity creates and that nothing had yet evolved to reliably solve. After everything, every animal that has ever lived, every nervous system, every immune system, every body complex enough to have a developmental program, a functional architecture, a lifespan long enough to learn and remember and change in response to experience. The ceiling lifted because the enforcement mechanism evolved. And with the ceiling lifted, the history of complex life on Earth began in earnest. The upgrade was not death. Death was always there, waiting for every cell in every organism from the moment life began. The upgrade was making death responsive, making it programmable, building into the cellular machinery a system that could receive information about the state of the cell and the needs of the surrounding tissue, integrate that information, and execute a controlled dissolution. When the calculation came out a certain way, that is what changed. not the existence of death, the existence of control over death. And that control, once it existed, became the foundation on which everything else was built. The embryonic development that sculpts a hand from a paddle of tissue, the thymic education that produces an immune system capable of violence without self-destruction, the continuous surveillance that catches damaged cells before they can become tumors, the neuronal pruning that refines a brain from overproduction to precision. All of it runs on the same underlying system. All of it is downstream from the same evolutionary innovation, the innovation of building death into the cell, so that the organism could tell the cell when to use it. Now consider what this means for the question the opening of this story raised. Why did life build death into itself on purpose? The answer, after everything this story has covered, is not that life made a sacrifice. Life did not sacrifice anything in any meaningful sense. Natural selection does not sacrifice. It selects. What happened is that cells carrying the precursors of apoptotic machinery survived better and reproduced more successfully than cells without it. When those cells were part of multicellular systems that needed coordination enforcement, over time, the machinery became more sophisticated, more reliable, more deeply integrated into the basic operations of the cell, because every improvement in its reliability produced organisms that held together better, that developed more precisely, that cleared damaged cells more efficiently, that maintained tissue integrity longer. The cells did not choose to die for the whole. The whole did not ask them to. What happened was simply that the systems which included cells capable of dying on cue outcompeted the systems that did not. And over hundreds of millions of years, that competition produced a mechanism so refined, so deeply embedded in cellular biology, that it now runs as quietly and as automatically as any other basic cellular function. You do not feel it. You have never felt it. You will never feel it. In the vast majority of the trillions of apoptotic events that occur in your body across your lifetime, the cell condenses and packages itself and disappears. And you are reading these words, completely unaware that somewhere in your body right now, in some tissue you will never think about, a cell is running the program that makes you possible. The darkest upgrade in the history of life was not death itself. Every living thing has always died. The darkest upgrade was learning to use death as a tool, to build it into the infrastructure of the cell, to make it responsive to signals rather than accidents, to transform it from the thing that ended life into one of the things that complex life cannot exist without. Death was always there. It was always going to be there. It was never going away. The upgrade was control, and that control, written into the genome of every complex organism on this planet, operating in every cell of your body at this moment, maintained across more than 500 million years of animal evolution without losing its fundamental logic, is what made it possible for the chemistry of a single cell in an ancient ocean to eventually become something that could sit in a chair and read about its own history and understand, for the first time in the 4 billion years that life has existed on this planet, what it had to give up in order to become what it is. Look at your hand. You have looked at it twice in this story. Once at the beginning, before you knew what you were looking at. Once in chapter 5, when you had enough context to see the developmental death written into its architecture. Look at it a third time now. With everything this story has given you, the space between your fingers, the 150,000 cells that died on schedule so that the fingers could separate and become distinct, that fact has not changed. But what surrounds it now is different. You know the molecular machinery that executed those deaths. You know the worm, with its 131 deaths, that never vary. You know the neurons that died, so that your brain could be wired with precision rather than abundance. You know the immune cells that died in the thymus, so that the ones that survived could protect you without destroying you. You know what happens when the dying stops happening correctly. All of that is in the hand you are looking at right now. The apoptotic machinery is in every cell of it. The caspases are loaded and waiting in each one. The BCL2 proteins are balanced on both sides of the mitochondrial membranes inside those cells, continuously updated with information about the cell's condition and the tissue's needs. P53 is scanning the genome in dividing cells, ready to tip the balance toward death if the damage it finds is too severe to repair. Somewhere in your hand right now, some cell is receiving a death signal. Some cell is condensing. Some cell is packaging itself for disposal, and the tissue around it is completely undisturbed. The cell simply gone, a space closing over where it was. Nothing marking its departure except the continuation of the function it was part of. This is happening in every tissue of your body simultaneously. Not a catastrophe, a maintenance, the quiet, continuous, invisible work of a system that has been refined across 500 million years of animal evolution to do exactly this. To keep the body coherent across a lifetime, in a world that continuously tries to make it incoherent. The hand that picks up a glass of water, that turns a page, that reaches out toward another person, is possible because of this system. The brain that generates the thought of reaching, the nervous system that coordinates the movement, the immune system that keeps infection from shutting both of them down. All of it runs on the same underlying infrastructure. The infrastructure of controlled death. The infrastructure that the ancient single-celled world did not have and could not have had, that had to be built from scratch over hundreds of millions of years through the only process available, and that now runs in you so seamlessly that you have lived your entire life inside it without once being aware of it. You are not a person who has a body that runs apoptosis somewhere inside it. You are the outcome of apoptosis. You are the shape that emerges when the system works correctly. The form that controlled death makes possible by continuously removing what does not belong to it. Every feature of your body that makes you recognizably human. Your hands, your face, your nervous system, your immune system, your ability to move through the world with coordinated purpose, was sculpted partly by what was taken away. The absences made you, and they are still making you right now in ways you will never feel and never need to. Life did not defeat death. Life learned to use it. And the hand you are looking at right now is the proof.